So I've been interested in the assembly problem for about a decade. Previously worked on short read assembly, and currently in the Abyss team, working with the Abyss team under Nanopore. This talk is not about a de novo genome assembler. It's about a scaffolder. It's not about error correction of nanopore reads. Instead, we have used this emerging long read technology to rescaffold draft genomes, high-quality draft genomes. So I'll be focusing on two aspects during my talk. The first part will be describing the method, and the second part will be giving you some examples where we've used this technology with long reads from different sources, not just Oxford Nanopore. Okay. All right. Okay, so there are some scaffolders that exist already. So why are we doing this? That will become evident during the rest of my talk. But some of the most popular scaffolders at the moment include S-Space Long Read and BB Jelly and AHA. And there are many scaffolders. Most of them are integrated in de novo assembly software, some of which are mentioned here. Most of those, all of those are alignment-based. In which you take paired end reads and align them to unitigs, quantic sequences, and order and orient. Did I miss a slide? I think I did. Oops. Okay, well, I guess I can go quickly over this, but scaffolding, essentially, what is scaffolding for those of you who don't know? It's one of the last steps of genome assembly, and it's a process by which you order and orient contig into a higher arrangement called a scaffold. And the goal essentially is to get one genetic element per scaffold if you can, such that you can make sense of the genome. Again, there's a really good resources out there because sequencing technologies have enabled the rapid and cheap sequencing of genomes. Unfortunately, a lot of these genome assemblies are fragmented, but they're out there, and now with emerging long reads technology, and some of which have been out for a while now, such as PacBio, you can actually use these long reads to rescaffold existing genomes. Okay, so yeah, so I was saying that most of them are alignment-based. The scalability and error tolerance of these scaffolders will depend mostly on the aligners that they use. So they may achieve variable successes on error-rich long reads, and I'll show this during my slides. Another point to keep in mind is when using paired end reads or MPET data for rescaffolding, you're limited to a library of a specific fragment size. So if you sequence many different libraries, you may cover a really wide spectrum and be able to increase the long-range contiguity of your assembly, but you're still limited by this. Okay, just briefly, one slide on this. For those who don't know, this is not commercially ready yet, but it's in early access phase. We've mostly worked with public data, so all that I'm going to show today is taken from public databases. This is a shift in sequencing paradigm. It's no longer sequenced by synthesis. Instead, you have a molecule, you force it through a pore, and disturbance in current is read, and a base call is determined. It's low cost. The sequences are long, about 5-6 kb in average, some of which are about 100 kb. We've seen yeast sequences, 150 kb. Some have reported up to 200 kb long reads. You end up with raw sequences, the template strand, the complement strand, and a combination of both called 2D or high quality sequences. The early chemistry from the vendor would produce reads with about 15% error. It's high, but what we've noticed from the data is indels in the data, which are quite high, tend to occur tend to cluster together, so they occur in stretches, which is a good thing for us. It's a good thing because out of the sequences, there are some really good k-mers, and this is an example, pairwise alignment from using an Oxford Nanopore long read to the reference E. coli data, and you can see the indels in the Oxford Nanopore read. And in blue, I've highlighted two good k-mers. So when you use them in pair, you have a specificity of being able to maybe or maybe not find them into your assembly contigs. You can use this information for pairing these contigs. And this was the rationale for developing LINX, the long interval nucleotide chimer scaffolder. It uses the scaffolding engine, which is from our lab, and also the predecessor to the space and the space long read scaffolder. Again, it's chimer-based. It's not alignment-based. You can explore a very vast chimer space. You're not limited by fragment libraries, fragment lengths from a specific library. It will work on long reads and draft sequences, virtually any length, almost any error profiles, except if you had, for instance, a occurrence of one base error per 10 bases, and you don't need to correct your reads before you're using it. Okay, so this is a bit of a complicated slide, so I'll try to go by step. Step one, you extract chimer pairs from your reads. You choose a distance interval between chimer pairs. That's the D parameter. And you use a step size to extract all the chimers. You store that in the hash data structure, seen on the right. Then you extract chimers from your sequence contigs and only store them if you've seen them in the reads. After that, you cycle through the first hash data structure and you locate the contigs. So I've indicated by these dotted arrows the contig numbers that these chimers are found in. And for all of those, you track the number of links, chimer pairs essentially, that have been seen between these pairs of contigs. And you can calculate an average distance or an estimated gap based on the location of the chimer and the contig, and the length of the contig as well. And the fourth step is to produce a layout. It's through an iterative extension where you look at every single contig and see its pairing partners. And that's the layout is determined by at least two main heuristics, and it's the number of links necessary to merge two contigs and the maximum links ratio between the second best and the best possible merge. Okay, some practical considerations for scaffolding. It works best if you run iteratively because you can explore a very vast chimer space. And you do this by increasing the distance between chimers and reusing the output from the previous iteration. Of course, you can imagine that storing chimers into hash data structures, and there's 3 of them, can use a lot of memory. It could be a problem. Memory is getting cheaper and cheaper, so you can rely on that, or you can change the step parameter. So if you increase T, the step parameter, you extract less reads, less chimer pairs. Of course, there's a drawback, and the drawback is that you diminish the space of chimers that you use for scaffolding, so potential loss of information here, which you can adjust by changing the number of links required for pairing contigs. I would rather not have to do this. In some cases, you'll see in some examples that for large genomes, it's sensible to do this approach. But another approach is to use a Bloom filter. And Bloom filters are space-efficient data structures. They're probabilistic, although in this case, that doesn't affect the algorithm whatsoever. And what you do is you take all your contigs from your assembly, you shred them into chimers, fill a Bloom filter, and then at step number 1 of the algorithm is you query the Bloom filter and see if these chimer pairs actually exist. And you can filter a lot of chimer pairs this way. You can imagine from Oxford Nanopore bio, where the error rate is substantially high, a lot of these chimers will be filtered out. Okay. So showing some results with the Oxford Nanopore, the public Oxford Nanopore data for E. coli, Salmonella typhi, and the yeast Escherichia. This is where you want to be. You want to have a really high contiguity when you scaffold or assemble, and very little misassemblies. I indicated in Chevron where the links rescaffolded assemblies are in this. It all started with draft illumina assemblies. Some were done with Abyss, some were taken from public websites. And we've rescaffolded with S-Space and AHA. These were the scaffolders. We also have for Salmonella typhi, and seen in red here, a Spades hybrid assembly that uses Nanopore as well. So in all of those, in terms of scaffolding, again, we're comparing scaffolders to scaffolders, not scaffolders to error correction. If you do correct the reads, some have shown that you can achieve essentially one scaffold or one chromosome to one scaffold. But some of the issues with these pipelines, they take a long time to run on 30x coverage, up to 5 days, where you can run links in minutes for 1 iterations, could be few hours for 30 iterations. Generally, the links assemblies will have less errors than other scaffolders, as seen here with S. cerevisiae. This is our lowest data point when you compare to this. Do I want to say anything else on this slide? Oh yeah. So one thing to keep in mind is, since it's a scaffolder, you can actually rescaffold assemblies that have been done by error correcting the Oxford Nanopore reads subjected to the cellular assembler. You can actually rescaffold that if you want. Okay, so this is for relatively small genomes, 12 megabase for yeast. Oxford Nanopore here used for both the yeast and the bacterial data set. Let's see if we can turn the knob a bit higher. This is, I'm going to show some results with arabidopsis. Again, this is public data, PacBio data that was released last year on the arabidopsis 120 megs genome. Okay, again, this slide is a bit complicated, but we had 2 assemblies. An illumina-only assembly, not sure what tool was used to generate this assembly, but it's orange, seen as the dotted line here. And we also have a null path assembly of illumina data as well, very contiguous assembly, 310 KB. When you use links on this assembly, raw PacBio data, only 4 iterations at a distance of 5 KB, 10 KB, 15, and 20, you increase contiguity of this assembly significantly. When you use EC tools, this is error correction tools for PacBio data from that same lab, you can pretty much double the NG50 on the original illumina assembly. The same pattern is seen here with the LPAT assembly using raw and error corrected, and with only 4 iterations of links. Cranking this knob a little bit more on bullfrog genome, estimated to be about 5 gigabases. This is something, some work in collaboration with Karen Elming at UVic, University of Victoria. In this project, what we've used is several data sets. We've used molecular data, we've used MPET libraries, and we've also used an assembly on a different K to rescaffold one of our assembly. Okay, so for this, we didn't have a lot of coverage from molecular, only 0.4x, but we did it anyways. As you can see, the NG50 using molecular did not increase all that much. Again, the library is 10 KB. We can go much further in distances in order to pair sequences. Our assembly was fairly fragmented to begin with, but with several rounds, so one round using MPET. And so what we did here, the trick for MPET is we generated pseudo reads from the MPET pair by filling it with ends and extracting chimer pairs at a specific distance between these 2 reads. And further rescaffolding using another assembly done at K128, we're able to triple the NG50 for both of them. Okay, so this is one of the largest genome ever sequenced, that white spruce. This is essentially to show scalability. So I have not been able to use existing scaffolder at the moment to rescaffold the spruce genome. We have some techniques in-house that work well on spruce, but one of our best methods is using a draft genome from a different white spruce genotype. So recently published is a version 4 of the P29. This is a Western Canada white spruce genotype, shown in red here. This was the NG50 for that version. And we used the blue one, which is the Eastern Canada pure white spruce genotype. And we rescaffolded with links 22 times using distances starting pretty much at 5 kb all the way to 100 kb. So you can imagine 100 kb, you're going to put a lot of ends between contigs if you find merges. This is all great, but is this valid? We don't have a reference for spruce. All we had was 3 MPET libraries, the largest of which 12 kb. And as you can see, these are all the iterations. At the 22nd iteration, we can still validate above 40% of these merges. We would not expect to validate all the merges because some of these gaps are large, very, very large. And this is not unexpected. This is the average gap size at 100 kb, about 40 kb, 40 kb gap size with a standard deviation ranging from 10 kb all the way to 70 kb. Okay. So some useful tips for scaffolding. Run links iteratively, increasing the distance, decreasing the step size. Create a bloom filter. It will be, with the current bloom filter, this will take some time on large genomes, but not on small genomes. And you see the payback as you do multiple iterations. You can also pre-compute it in the distribution. There's a tools folder. You can actually use a script to do this. And reuse the filter at each iteration. Okay, so in summary, I have just 3 things to say. This is a K-mer scaffolder. It's alignment-free. You can use varied read sources, error-rich long reads, corrected long reads, draft assembly, and it's scalable. Some of our future work will include developing a new, faster, more efficient bloom filter, and also we intend to port this code to C++ to make use of parallel libraries and make everybody's life a bit easier when running on many, many reads. I'd just like to finish by thanking you for listening, and also thanking Inas Brual for allowing me to work on this project in his lab, the John Number Knowledge Fund for covering part of my travel to this conference, and also received funds from Intel, Genome Canada, Genome BC, the BC Cancer Foundation, and the NIH. Thank you very much. I'll take any questions.
Thank you. So, any questions?
Very nice work, and I have a question. Maybe I'm misunderstanding, but I'm curious on. Usually, in the de novo assembly, we use assembler, especially on the de novo graph-based assembler. They may use very long K-mers for assembly, and due to the very high sequencing error of the long reads, you may use shorter K-mers. So, what will happen if the short K-mers match to, for example, multiple contigs?
Right. Yeah. We filter those out. We actually track the multiplicity of K-mers. I didn't mention that, but this is tracked in our data structure. So we only pair contigs if these K-mers are found uniquely in the contigs.
However, if the distance between the multiple K-mers along the long read are similar, match to different contigs, what will happen?
Yeah, like I said, like the K-mers, if a K-mer is found multiple times, so if you have a pair where you have K-mer I prime and I prime, and this one also pair with I prime prime prime, we essentially don't use it. We don't use that pairing. Yeah.
Okay, thanks.
Yeah. You're welcome.
So what's the typical K value you use?
Okay, yeah, that's a good question. With Oxford Nanopore, we found that a K of 15 was ideal. Okay. And with spruce, we used 26. So we did some assessments before using these K, and I would encourage people to do the same. In the distribution, there's a recipe to redo all this analysis that I showed here. So all of this is reproducible. Just download links and do a run all on the distribution in the test folder.